Okay, so for today's topic, analysis of single-cell sequencing data by CLC Bio and Ingenuity, we're delighted to have our speaker Naomi Thompson. And Naomi Thompson is currently the Director of Clinical Analytics at QIAGEN, and she works with bioinformatics and the launch of the Gene Reader. Naomi joined QIAGEN through the acquisition of CLC Bio. Before that, she was engaged in many roles in DNA sequencing and DNA sequencing analysis, including the oncology department at the Bristol Myers Squibb. Naomi received her education in biology and business from Brian Mulroy College and LaSalle University, respectively. Okay, without further delay, I would like to introduce you to our speaker, Naomi Thompson, and her talk, Analysis of Single-Cell Sequencing Data by CLC Bio and Ingenuity. Naomi, please go ahead.
Thank you, Lily. Good morning, everyone, or good afternoon. I'm going to be continuing a bit on our series from QIAGEN about single-cell analysis. I hope you were able to attend the seminar that was given 2 weeks ago on a Wednesday at about this time by Krishnam Alapalam, and that focused more on the pre-analytic aspect of the single-cell analysis, and we're going to focus more on the post-analysis stages, but we'll also spend some time reviewing a little bit about what was presented in the initial seminar, so you'll have that foundation. So the first question one might have is, why would you do single-cell analysis? And there are a variety of reasons that one would do that. And if you think about it, cellular populations, like all populations, are somewhat heterogeneous. And if you were to, for instance, be surveying a kindergarten class and you gave all of the kindergarten class an exam, you would get a different kind of information if you take the norms and averages of how the children performed in the class versus the data that you would get if you were to look at the result of each student's exam. You might be able to identify students that are targeted for accelerated classes versus those that might require special help to advance them. And the same thing is true for cellular populations. If we're looking at a cellular population and we look at the average, we're going to get one kind of information. And if we're able to do more specific analysis of each of the individual types of cells, then we'll be able to get more specific information about those cells. So, and this is particularly true when one is looking at the data that one gets from cancer cells. Cancer cells are known to be heterogeneous. They're heterogeneous in terms of single nucleotide variation, copy number variation, expression, and these are all types of differences that we can measure with next generation sequencing and then analyze with bioinformatics. Another reason that one might want to focus on single-cell analysis versus a bulk analysis is that when you're able to do single-cell analysis, you're able to take advantage of the fact that your sample DNA is so precious. You may not have large volumes of DNA. The types of samples that you might be working with might be circulating tumor cells, cells from small biopsies such as a fine needle aspirate, or you might be taking cells from in vitro fertilized embryos at very early stages, in which case the amount of DNA material that you have is also very precious. And sometimes one is able to overcome that by cultivating the cells and growing them up, but you're always concerned, particularly when you're looking at expression analysis, that that cultivation process is going to change the behavior of the cell and not truly reflect the in vivo environment of the cell. So one wants to be able to really focus on an individual cell, understand the unique characteristics of that cell, and eliminate results that are based on an interpretation of average behavior of a large group of cells versus the behavior of an individual. The other is also useful, but it's important to know both. So what we have illustrated at the top of this slide is the what one might see as typical tumorigenesis, where you would start with normal cells and then you would have the introduction of a mutated cell, and over the course of time you would find that the nature of the populations of cells are going to change as different mutations are accumulated in different cellular populations. Some of the mutations would be driver mutations, some passenger mutations, but all of them are characteristic of the different cells that could be in primary tissue, secondary tissue, or all part of a single biopsy. And this would lead to additional cell growth. And if one is using information about those cells to direct treatment, it's important that you have a strong understanding of the individuals. And this will become even clearer as we are looking at the results that we have for the population of cells that we're looking at in this single cell analysis. So what are we getting from QIAGEN? And what you are getting from QIAGEN is a true source for all of the stages in a single cell analysis, starting with the ability to amplify your DNA using the Repligy single cell kits and the quantumized kit for understanding the quality of your DNA. And if you're doing an RNA-seq analysis, making sure that you have eliminated the genomic DNA when you're doing and also making sure that you've eliminated the ribosomal RNA that can really interfere with high sensitivity detection. We also have targeted enrichment with the gene-read DNA-seq panels, and this is coming from the acquisition that QIAGEN made in Frederick with the Assay Biosciences. We then have library construction with the gene-read library preparation kits. The NGS runs can be run on any sequencer, so if you have a next-gen sequencer in your lab, you can use that. You can also learn more about the gene reader sequencer that is part of the complete NGS sample to insight system that we launched at the molecular pathology meeting in November. And then, again, part of QIAGEN's recent acquisitions are the data analysis and interpretation tools that we have from CLC Bio and from Ingenuity that provide a great deal of power to the user and insight. So I'm going to now go through each of these steps that we highlighted here as part of our whole overall system one at a time, starting with sample isolation. And so what we're looking at here is a very specific method. Sorry, I just got a notice that the broadcast is now starting. So I'm going to do a quick backup, a little bit of what we've covered so far. That we're going to be going through an overview of the sample to insight for single cell analysis, that we're going to be reviewing the techniques that will focus on the advantages of doing single cell analysis, specifically looking at single cells and getting this unique information from single cells and overcoming the bias that one has when looking at a population of cells. And I just went through the advantages of QIAGEN sample to insight, offering from sample isolation to interpretation and leveraging all of the internally and externally and acquired technology that QIAGEN has brought together to provide a real solution for our users. And I was just about ready to talk about the specifics of whole genome amplification. So if you missed anything, this is where we are right now. So whole genome amplification is one of the areas that QIAGEN has unique capabilities. The whole genome amplification is based on our Repligy technology or Repligy Sensify technology, and it's unique compared to other types of analysis. For instance, if one is using PCR-based technology, which would be the alternative to Repligy, you would be first starting potentially with a degenerate oligonucleotide, or perhaps you would be using a primer extension for preamplification, and this would all be methods that you would use to get more DNA for doing your downstream next-gen sequencing. And all of those would be based on the Taq polymerase. Taq polymerase has a limit of about 3 kilobases for making PCR strands. It's known to have a large number of errors and an amplification bias because you're constantly cycling through high temperatures and low temperatures where you're annealing the DNA template and priming, and it has known issues with secondary structures because of this constant priming and amplification. So, and very often prior to doing a PCR, you might start by creating an Epstein-Barr virus transformed cell line to even get enough DNA for your starting point. And this is in direct contrast with the multiple displacement amplification on which the replogene system is based. In this case, you are starting with a random hexamer followed by strand displacement synthesis with a 529 polymerase. This has no problems with the branched DNA structures that it creates and is able to generate fragments that are over 100 kb in length. The 529 polymerase has known proof rating, so it has greater than 1,000x times fidelity than the Taq polymerase. Part of this is because the polymerase is able to stay associated with the genome template. It's an isothermal amplification that's taking place at 30 degrees centigrade. Really minimizes the sequencing bias, and it's further facilitated by all of the optimization that was done and is provided to you with the QIAGEN kit. So you're able to start directly from cells, or tissue. You can work with DNA or RNA, and the result will be either an amplified cDNA or an amplified DNA that would be ready for your downstream amplifications. And you can store this without any degradation. It's a huge advantage, and this is part of what we'll be looking at some of the case studies that we see later on in this presentation. The next step in the process, once you've generated your DNA, you've now got genomic DNA at an amplified volume so that you can move forward and target the region that you're most interested in. So this would now be taking over at the point that you would have if you had not had such a precious sample, or if you weren't doing single cell analysis. You can use the DNA that you generated from the replogene with the GeneRead DNA-seq targeted panels. And there are 15 different types of panels that we have for different tumor types. They can start with as little as 10 nanograms of DNA for the hotspot panel. And these are very targeted amplifications. And the advantage of that is you then are able to get the depth of coverage that you need for these very sensitive low-frequency variant detections. If you are doing whole exome or much larger panels, what you'll find is that you're not going to get the depth of coverage, or it will be very costly to get the depth of coverage that you need for this kind of sensitivity. And QIAGEN has gone through the process of understanding the genes that are associated with each of the cancer types and targeting the known regions that would enable you to have the best insight. So now I'm going to start to focus on the analytics, which is, as I told you before, I'm coming from the CLC Bio. The purpose of this talk is to really focus on the bioinformatics, and I'm going to spend a lot more time here. And the view that you're seeing right now is a subset of screenshots from the biomedical workbench, so you can get an idea of the power of the tools that will be provided to you with this software application. You'll notice on the left-hand side that you have a large number of ready-to-use workflows, and they're organized based on the type of analysis that you would be doing. So you can have analysis for identifying inherited variants. You can also have for somatic variants. There's specific analyses for targeted amplicon sequencing, and in fact, we have a workflow for each of the GeneRead panels that I mentioned to you before for each of the cancer workflows. We also have, in addition to the already created workflows, we have a number of tools that you can use and build on your own. So in addition, we provide you very useful out-of-the-box workflows, as well as the ability to tweak and create your own tools. And what you see over here on the right side is fraction of one of the workflows that we've put together for you, where you can start with a workflow input such as a FASTQ file, and one of the tools in the toolbox, this is one for mapping the reads to reference. If I were to click on this, I would be presented with a variety of parameters that I could use to optimize this. For your advantage, these parameters have already been optimized. And then once you generate your read mapping, you would then go to the process of doing a local alignment. And this would proceed automatically once you just clicked the start button. One of the first things that you would want to look at after you had done an analysis is to know how successful was your analysis. So we provide you with in-depth QC data. So what we're looking at here is the kind of coverage distribution that you have, and you can see that you have a large amount of distribution of coverage. The majority is in this 0 to 1,000, but you also have the 5,000 to 6,000, 10,000 to 11,000, all the way through the to some areas where you have coverages as deep as 65,000. And then you also can find out what percentage of your data is covered. So at least 98.94% of your coverage is present at 1x, and 95% is covered at least 100x. And then you can also see for each target that you had specified in the gene read panel what your level of coverage is and the specificity. So really a great deal of information to help you know that the targeting that you did in the previous part of your experiment was successful. And then another key attribute of the bioinformatics is we're going to enable you to review the history, so you can always know exactly what types of analysis that you did. So if you decide to modify any of the workflows and look back at your results, you can see what the analysis was that you performed and how you might have modified it in future. And one of the most useful and where you spend the majority of your time within the bioinformatics tools is the visualization. And in this case, what you're seeing is your reference sequence at the top. So this is the reference that you mapped your reads to. Then you can see any amino acid changes that have taken place. This region here is your mapping, and you'll notice that we are not displaying hundreds or thousands here, even though there's 12,591 reads here. You'll see a representation of the individual reads in the top half, and then a compressed version of your reads in the bottom half of this graph, so you can see specifically where your variant is showing up, and also how well it's represented in forward and reverse reads and the greater depth of coverage. And all of these variants are linked to a variant table that you can use to navigate from one variant to the next after you've done your variant calling. So an incredibly useful tool. And we additionally provide you with bioinformatics that allow you to focus uniquely on the kinds of challenges that you might be faced with when working with an amplified sequence from single-cell analysis. So what we're looking at here is all the information specific to this single-cell analysis and this targeting. So we're looking at one of the gene read panels, the 101X clinically relevant tumor panel. And this is showing you the region that you've targeted, as well as all of the primers that were used to create the amplification. And when you're looking at the reads below, you can now see those reads that are in the reverse orientation, those reads that are in the forward orientation, and you can also see paired end reads. So here we're seeing one side of a paired end read, and you can see that part of the paired end read, which includes this shaded area, and this shaded area maps to these primer regions. So you can tell that the primer region here is included in the mapping, so you get the greater specificity of the read mapping, but is not included in the variant calling. So we are not artificially directed to look for variants where the primers are. And this is based on this specific functionality that is allowing us to trim primers and their dimers for mapped reads, and that's what we're seeing here. This is a primer dimer here. One of the other characteristics that one can't emphasize enough is the quality of the algorithms that one has when you are working with the genomics workbench and the biomedical workbench. These are world-class algorithms that were developed at our site in Aarhus. And we, in my saying that is typically not enough to provide you with the information that you need to have confidence in that. So we've done a number of benchmarking assays, specifically with the NA12878, or the genome in a bottle reference sequence, which has been very well characterized. So we're able to make statements not only about the true positives, but also about the true negatives, and give you some statistical information about how accurately our algorithms are able to work. So what you see here is information about sensitivity, specificity, precision, and the false positive rate. And you can see that sensitivity here, which is the number of true positives divided by the true positives and the false negatives, is very high, and our specificity is even higher. So the true positives is telling us if a variant is there, and are we able to find it, and the specificity is telling us the kind of the opposite of a false positive. This is telling us that we're finding, or we're not calling variants that aren't there. And the precision is telling us of all the variants that are called, how many are truly positive. And then you can see here the advantage of getting coverage. So here we're looking at sensitivity and the false positive rate, where the light blue bar is telling us our sensitivity in regions that don't have this minimum threshold of 200x. And the dark blue bar does have that cutoff. So you can see there's a definite advantage in making sure that where you're calling your variants is it in an area that you have appropriate coverage. And this is one of the criteria that you can set within the biomedical workbench, and one of the advantages of using the gene read panels, because they ensure very good coverage at the regions that are important to you. So one of the things that we're able to provide to you at QIAGEN is all of these pieces together. So we, as we've mentioned, are platform agnostic, but we also have even more streamlined and more holistic application from sample to insight when we're using the gene reader, but we can work with any sequencer. We can use the gene read panels, perform the analysis with the biomedical workbench, and then use an interpretation from the Ingenuity. And this gives me an opportunity to tout the knowledge base that we have access to from Ingenuity. This knowledge base is the foundation of Ingenuity pathway analysis, which has been the gold standard for doing expression analysis and pathway analysis for over a decade. Excuse me. The knowledge base provides a breadth of biological and chemical information that allows us to really see the relationships between those pieces of information because of the way that we have organized it. So you're able to see information from an individual and use that to understand their phenotype. And there you might be able to understand information about drug mechanisms and associate the drug mechanism with a cellular mechanism. And that cellular mechanism can then further be associated with a molecular mechanism. And that molecular mechanism can then be tied to a very specific RNA or DNA alteration. So the DNA alterations, or the SNVs, would be what is highlighted in Ingenuity Variant Analysis, and the differences in expression are highlighted in Ingenuity Pathway Analysis. But the real value that we're providing is, in addition to having highly accurate, highly curated data, is that we've put them together in a structure that allows you to have access to the content and relationships and take full advantage of the investment that QIAGEN's made in this ontology. And then when we put that all together in a single-cell analysis, you're able to see that the biomedical genomics workbench is going to take you through a certain point where you can take your data off of the sequencing instrument, align it to your reference sequence, generate variants, and then once you have identified all of your variants, you can now upload that to the Ingenuity Variant Analysis. So the point of being in the biomedical workbench is to identify true variants. And then once you've uploaded those into Variant Analysis, you're able to put that through a cascade of additional filters that will allow you to identify those that you have higher confidence in, those variants that you may not be interested in because they are common, such as those that are present in dbSNP or in our allele frequency database, which we are curating. You can also identify the variants that have known deleterious effects, and then use some association of the known deleterious effects with genetic information, statistical associations with diseases from the curated literature, and further associates that within a biological context. So if you know that this particular sample is coming from a colorectal cancer, the variant analysis will take advantage of that information to further filter and provide information about the relevance of the variants that you've uploaded. And then this type of analysis is possible to do at a variety of scalable workflows. So you can use this for a large number of samples, or you can use this for an individual sample. So you can use it for stratification studies, large cancer studies. You can use it for a genetic disease cohort. Typical example might be for trio analysis or other types of family analysis, tumor normal pair, or a personal genome. And for obviously, when we're talking about single-cell analysis, it would be to your definite advantage to use this to analyze common variants or shared variants amongst cells that are coming from the same biopsy, and those things that are different, to be able to know what's unique and what's shared, what are possible driver mutations, and how the populations may be changing over time. So really powerful tools that you can store and share with other colleagues. So now that I've spoken about the strengths of the tools that we're providing, I also want to spend some time talking about the two case stories that we have. And in the first example, I'm going to go over how we use the bioinformatics tools to illustrate the strength of Repligy versus Malbac, which is another method, a PCR-based method for doing the whole genome amplification. And then we're going to use the analysis tools again to look at single-cell analysis of two different colorectal cancer cell lines. So in case story 1, we're focusing on Repligy versus Malbac. And here you can see we're doing two whole genome amplifications, and that's really the only place where these two different patterns differ. On the left side, we're targeting with E. coli, just 1 picogram, and we're using the Repligy single cell kit, using that to, with the gene reader library kit, as in the other, to create a library. And then we're using the MySeq sequencing and the 2 times 150 nucleotide version 2 amplification, and then the biomedical genomics workbench for the data analysis. So again, to emphasize, exactly the same for the two different workflows. The only difference is whether or not we use the Repligy single cell kit or the Malbac kit. And just to tell you a little bit about Malbac, this is a multiple annealing and looping based amplification kit. It was developed at Harvard. It's based on pre-amplification prior to an exponential PCR. It was developed at around 2012, and it is PCR and TAC based, as opposed to the whole genome amplification method that we described earlier for Repligy, which is isothermal genome amplification. So everything there is taking place at 30 degrees. The first thing we'll do is we'll look in detail at the read mapping, and I'll explain to you a little bit about what you're seeing here. So again, at the top, you can see your reference sequence, and we're looking at two different read mappings simultaneously. So this is in our genome browser, and you have a lot of flexibility in how you're able to configure and put data together. Here we have the E. coli reference sequence, and you'll notice we're not looking at each of the individual bases. We've turned on an option so that only the bases that differ from the reference sequence are showing up here. So you can see we have an inserted T here, we have an A here. These are paired reads, so you'll notice you have a K here, which is representing a G and a T, because these paired reads were put together and they had equal quality values for both bases at this position. So you'll see we have the T base, which matched the reference sequence, and then we also have a G, which did not, so we're flagging that with this K. You'll also notice that we have these individual lines here, which is showing you where the pairing has taken place. So you've got this read, and they are known to be paired with this link here. In cases where the pairing is completely overlapping, that's when you'd have this synthesis. So this is for the repligy, and you'll notice in this area we have 3 different variants. And you look at the exact same area in the Malbec generated single cell amplification. And here we have many, many more variants, including some multi-nucleotide variants, showing a much greater level of diversity between the two. And since this is a known sequence, we know what the reference sequence is. That's why we chose E. coli. We're able to say that this sequence is correct, and so there should be no variance, and yet we're seeing many, many more in this Malbec generated analysis. The other thing that's really important, and we mentioned before, is that it's important to have even coverage across the entire genome. So in some cases, an analysis might tout that it has very high average coverage, but average coverage is not good if it's very uneven. So here you can see that we have a maximum coverage of 3,000. The same thing is true here for Malbec, but we have very uneven coverage. We have some areas where it's very low and some where it's much higher. And this is at a very zoomed out level, so you can see across the entire genome. And here we've zoomed in a little bit, so you can see in the overflow region that you have much more even coverage. Here you don't even have any of the overflow coverage in this region of the genome, so it's much less than you have here. You might be curious about why we have these yellow reads here. Those are reads that have the potential to map in other locations in the genome, so those are just flagged as part of our information about the read mapping. And again, the blue reads are those that are present in pairs, and the red and the green are representing the red is the reverse and the green is the forward. This is showing you the different zoom levels, but the take-home message here is that you have much more even coverage with the repligy than you do with the Malbec. And so the summary story is that if you're looking in total, repligy versus Malbec, the number of false positive mutations is much, much lower. We only had 6 insertions for repligy, whereas with the Malbec, we had 231 different types of mutations, 222 SNP/SNPs, 6 deletions, and 3 insertions. So completely different type of error profile. And we also saw that we had a much higher of the percentage of the bases mapping for the repligy versus the Malbec. So the next case story is focusing on 2 different cancer samples. These are cancer samples that are from 2 different colon cancer cell lines. So, and we're again taking advantage of QIAGEN's sample to insight. The only piece here that is not coming from QIAGEN is the single cell isolation that is coming from cell raft arrays, cell microsystems. But thereafter, we're starting with the whole genome amplification, which again is the really key factor here for generating quality whole genome amplification with the repligy single cell kit. And then we're doing target enrichment with the human colorectal cancer gene read DNA targeted panel 2. This is going to amplify 34 genes that were selected based on our understanding of carcinogenesis and colorectal cancer. And then we're going to do the library preparation and sequence on a MiSeq. The data analysis is again going to be done on the biomedical genomics workbench with the data interpretation on the Ingenuity variant analysis. And we're further going to take advantage of QIAGEN's expertise with pyrosequencing using the PyroMark Q24 assays for confirming some of the known pathogenic variants that would be identified in each of these cell lines. And the for each of the cell lines, the LOVO and the HT29, we're doing 6 different samples. The first 2 are bulk samples, one that is done as a standard bulk sample and the other that is done as a bulk sample using the whole genome amplification to see if we're getting any bias other than the bias that we might introduce with single cell amplifications. We want to make sure that the type of results that we get from the bulk analysis is similar to the type of analysis that we get with the bulk whole genome amplification analysis for both cell lines. And then we're going to drill down and see what kind of unique and individual characteristics we can identify in each of these single cell lines that were isolated. So here you can see the first results. So what we've done is we've taken each of the key variants that we've identified and plotted them based on their allelic fractions. So we can see we have some here that are present at a very high allelic fraction and some that are present at a low allelic fraction. And these are the alleles, and we can see that for the most part, the alleles that are identified for the bulk DNA are very similar to the ones that we have identified at the same allelic fraction for the whole genome amplification. So in general, we're finding that we're not introducing significant bias when we're doing the whole genome amplification, and we're ready to then take a look at using this as a method for measuring the individual cell lines. And so here we are looking at, again, each of these individual cells. Well, again, here's the bulk and the whole genome amplification for the bulk, and the 3 individual cell lines for the LOVO-3, as well as the same below for HT29 for whole genome amplification. And here we're looking at the KRAS G13D mutation, and here we're looking at a BRAF mutation. And using the DNA sequencing, we're looking at an allelic fraction of 74% with our sequencing method, and this is confirmed with 71.37%. And you'll see when we do the whole genome amplification, we have 73.3% and 67.82%. Again, for the KRAS mutation, and we'll see even tighter mapping for the BRAF mutation, where we have almost 25% and 24% here, and 28% and 28% here for the sequencing detected versus the pyrosequencing detected variants. So we're very confident that we've not introduced any bias for the whole genome amplification. So then we can take a look and see how different are the single cells. So here you can see that the percentage or the allelic fraction for this individual cell for this KRAS mutation is 67%. Here we're in the 90th percentile, around the 59, 55 percentile. But again, you can see that the single cell amplification is consistent and confirmed by the pyrosequencing. So we have really good evidence that this is a great way to characterize the individual characteristics of single cells as opposed to a bulk amplification where you have completely different understanding of what the characteristics of the single cells are. And you can imagine that would be particularly useful when one has data coming from different tissue, primary tissue versus mutagenic sample, or metastatic sample. And once you have identified your cluster of variants, the next thing you would want to do is upload all of the variants from each of the experiments and see how those are identified within variant analysis. So here you're looking at the chromosome on which the variant takes place, the position, the region, so here we're looking at exonic regions, the gene symbol, the protein variant name, and if there's multiple transcripts, you would see multiple transcript names, and then each of the case samples. So here, each of the blue areas is showing you that you have a loss of function. Each of the orange is showing you a gain of function. And each of these bars here, as we have on the left, the ones that are associated with the KRAS mutation and the LOVO cell line. And on the right, you have the HT29. So here you can see, here's the BRAF mutation, here's the KRAS mutation. So you can see we had the BRAF mutation is the one that is on the right. So that's the HT29 cell line over here. And for the KRAS, we have the mutation that was present in the LOVO cell line. And you can see that this was heterozygous here. So a great deal of information is available for each variant, and you're able to see how each variant is represented across the case samples that you're looking at. And it allows you to know, based from a variety of pieces of information, what the impact of that mutation is. And clearly you would see a lot more here if I was able to expand this table, which you'd be able to do when you're using this, but this will tell you based on SIFT and polyphen and conservation scores if this is predicted to be damaging and how well it's conserved. And all of this can be used to help you to interpret the results of the variants that you've identified. And here you can also see the allelic fractions that were identified in the this region is also uploaded as part of your data into the variant analysis. So you can capture all of that as well. Furthermore, anything that you see that is describing the impact, if it's associated with a given publication, you can also drill down to that publication. So you'll have links here from PLOS ONE, from Digestion, and you can click on these links and see the abstract for these findings. So, in summary, and hopefully leaving you still some time for some questions, what QIAGEN is able to provide to you is a full sample to insight that's focused on all of the challenges, particularly the challenges of introducing bias in a single cell workflow. The results are ensuring quality at each step along the way. We were able to show you two different types of analysis that are leveraging the QIAGEN's kits and QIAGEN's bioinformatics. So you could see both the advantages of using the Repligy versus alternative PCR-based methods for genome-wide amplification. And you were also able to see how you can leverage our bioinformatics tools to first understand the variants and see the variants that you've identified, and then to further take those variants into a well-curated, highly accurate knowledge base so that you can see the impact of the variants that you've identified. It's a really powerful set of tools, and it really empowers people who aren't familiar with bioinformatics to use their genetics or oncology background and not spend the majority of their time trying to learn command line or other tools that would take away from their ability to spend time understanding the fundamental nature of the experiment. So you'll see that with using the Repligy kit, you're able to improve your amplification bias, get even better results, and you're able to normalize variant frequencies across a larger region, and you're able to eliminate bias of variants because of longer stretches, and able to identify contamination and help in normalization of variant frequencies. So I want to now thank you very much for your time and give you an opportunity to write in questions, or you may have written in questions as well already.
Thank you, Naomi. Thank you for giving this interesting topic. And I apologize again to our attendees who have the problem of accessing the webinar in the beginning. And now we have 5 minutes left over for room for some questions and answer sessions. The first questions I would like also to address here is, Naomi, since you mentioned a lot about the CLC bio and also Ingenuity, how do they really work together? If you could give us some brief introduction.
Oh, it would be my pleasure. So CLC Bio and Ingenuity were acquired with Biobase, actually, around the same time in 2012, 2013 by QIAGEN. And since that time, even though we as individual companies were also working together, when we were put together with QIAGEN, we've done a number of things to ensure that the handshake between the applications is very strong, as well as making sure that we're supporting the data. And this is true not only just for the bioinformatics, but also for gene read panels, and even more specifically for the gene reader. So we are able to put together an entire system that is compatible all the way across the experience that the user has from the time that they're extracting their DNA all the way to the interpretation of the result. Does that answer the question? Hello? Have I lost everyone?
Yes.
Okay, sorry.
Yes, Naomi, I'm here. I think I just muted myself.
Okay.
Yes, I think that's an interesting to know that. And I have a second one just to address that during the last 3 minutes. Since you've been also busy and engaged with the gene reader, would you like also to introduce or give a bit of talk on how does then the CLC Bio and Ingenuity connect the gene reader?
Sure. It's actually a very tight association, starting with the knowledge base, which I mentioned is a huge resource for our customers and for QIAGEN. We took advantage of that to design a very specific panel. It's the Actionable Insight Tumor Panel. And the Actionable Insight Tumor Panel really focuses on the variants that we have known information about. And then we're able to use that to ensure that all of the reagents, all of the user's time is focused on identifying variants that they can then use for further interpretation. And so we're using the Actionable Insights Tumor Panel. We've then optimized all of the bioinformatics to work with the gene reader data so that it will provide QC information, telling you exactly how effectively your experiment has taken place, and you never have to worry if your experiment hasn't worked well because the parameters weren't set up correctly. They're all optimized to work perfectly. So the only thing you would do for QC is perhaps my sample is too degraded, or perhaps I had an error in my workflow, which the QIAGEN support would be able to help you work through. But it's really set up to be a very easy system, and at the end of the analysis you will get a report that describes all of the variants that you have identified and links them together with findings, including clinical trials that might be associated with that variant, a rich bibliography that's associated with that variant. And when you're creating the report, you have the power to decide what information you want to include and what you just might want to review during the review process.
And that sounds like a very powerful tool actually we're giving, right?
I think so. It was certainly well received at the molecular pathology meeting.
Yes. Okay, so well, I think that's all for today because we only have 1 minute left. Thanks again, Naomi, for today's talk, and thank you for everyone for joining us online. And I hope you will find it useful. At the end, if you have any questions you would like to ask us, feel free to email us at QIAwebinars@QIAGEN.com, and we will follow up your questions personally. And also, if you would like to listen again to today's webinar or invite a friend to listen, and the recordings then will be available at the on demand shortly. Thank you very much for joining us, and bye for now.